Hi everyone, back to channel. Today we are going to start about endocrine system histology. So we know endocrine system is nothing but which directly pours out the hormone into a blood, into circulation. So it mainly involves in control of metabolic activities and also internal environment of the body. And this endocrine tissue are mainly highly vascular and also they have a secretory pole, that is nothing but secretory part. These are ductless glands because they doesn't require any duct directly pour into blood. So towards the wall of a capillary mainly, because through blood only it need to enter. So in the capillary wall they have their secretory pole, and in exocrine glands the secretory pole is mainly towards the surface over which the secretions are discharged. And next let us see the endocrine cells are mainly distributed into three different ways. So what are those? Like some cells are entirely endocrine. They are known as endocrine glands. For that examples are also known as ductless glands. So for this example you can take Hypophase Cerebral, Pineal Gland, Thyroid Gland, Parathyroid, Suprarenal, which we are going to learn now in everything in a detail. And next there are also some of endocrine cells which are also associated with some other functions, which is not mainly for endocrine, that means not for only producing hormones, but they also have some other functions. Then that kind of cells examples are like islets of pancreas, interstitial cells of testes, follicles, corpora lutea of our ovaries. So all these are having also other functions rather than being an endocrine gland. And also they are saying that liver is being mainly partly an endocrine gland, not completely, just partly an endocrine gland. And next, isolated endocrine glands are also found mainly typically in the gut, also in endocrine cells of the gut, and also similar cells are also present in our epithelium of respiratory passages and also other locations, like they produce amines mainly. Some are showing that the amine producing are also known as having endocrine functions. So many of these amines also are acting as neurotransmitters and also neuromodulators. So these widely distributed cells together now known as neuroendocrine system, or also known as APUD cell system. So this also we are going to learn in detail. And next, so on the basis of the chemical structure and hormones are mainly belonging to four main groups. So first one is amino acid derivative hormones. For that examples are adrenaline, noradrenaline, thyroxine. And there are also small peptides like encephalin, vasopressin, thyroid regulating hormones, and proteins example are insulin, parathyroid, thyroid stimulating hormones, steroids example are progesterone, estrogen, testosterone, cortisol. So these all things we know. Now let us start with the Hypophysis cerebral. So Hypophysis cerebral is nothing but a hypothalamus. The hypothalamus gives it neurons actually downward to the pituitary gland, that is anterior pituitary. You can see this is the pars anterior pituitary, and the posterior pituitary, and this is the median eminence, and this is the mammillary body, and this is the optic chiasma, and this is the infundibulum, and this is the posterior pituitary, and this is the cleft. Pars intermedia is the one which is dividing the pituitary into anterior and posterior, and this is the pars tubralis which is belonging to pars anterior. So remember, pars anterior has three parts, that is pars tubralis, pars intermedialis also belongs to pars anterior, and remember pars posterior pituitary is in direct connection with the hypothalamus or brain, you can say. So posterior pituitary is also known as pars nervosa because all nerves also enter into this posterior pituitary. So Hypophysis cerebral is also called as pituitary gland, and the floor is present in a third ventricle of a brain, and it is having a narrow funnel shaped stalk called as infundibulum. So narrow funnel shape is called infundibulum and lies in a depression on the upper surface of the sphenoid bone. So this pituitary is present in the upper surface of a sphenoid bone where it has its depression. And next, its own activity is also influenced by the hypothalamus, right? So pituitary is nothing but influence, that is it is under control of our hypothalamus and by also pineal body. So what are the subdivisions of Hypophysis cerebral? As I said, pars anterior, pars intermediate, pars posterior, also known as pars nervosa. And also there are pars posterior contains numerous nerve fibers, as I said, continuous with the central core of a infundibular stalk. So it is made up of nervous tissue, and also two parts, and a pars posterior and the infundibular stalk are together called as neurohyperfaces, because infundibulum from where the neurons are coming and the pars posterior. So infundibulum, pars posterior together called as neurohyperfaces. So the area in the floor of a third ventricle, that is also known as tuberculorum, is immediately adjoining the attachment to the infundibulum. Then it is called as median eminence. So this is the median eminence. So some say median eminence is also under neurohyperphases, that is our posterior pituitary. And next, the pars anterior. The pars anterior is also known as pars distalis, and pars intermedia are both made up of cells mainly direct secretory function. So they are collectively referred to as adenohypophysis, also anterior pituitary. So the extension of the pars anterior is surrounded by a central nervous core, also known as infundibulum, right? Same like this, it also have infundibulum, right? And because of this tubular shape, the extension is called pars tuberalis, and the pars tuberalis is a part of adenohypophysis. So pars tuberalis, pars intermedia, all these are part of pars anterior, and pars posterior parts are infundibulum, median eminence, and also its continuation as pars nervosa. So this is all about anterior and posterior pituitary. Now let us begin the histology of the adenohypophysis. Now you can understand adenohypophysis means pars anterior. So the pars anterior consists of cord of cells separated by the fenestrated sinusoids. So we can see this is a pars anterior. It is containing cords of cells, right, and it is separated by, you can see, fenestrations, that is nothing but sinusoid gaps. And there are several types of cells, mainly production of different types of hormones over here. And pars anterior is divided mainly into chromophile cells, and that have a brightly stained granules in the cytoplasm. So chromophile cells are having bright stained cytoplasm, and these and also there are chromophobe cells. So phil means bright stained cytoplasm, phobe means what, in which the granules are not prominent, means like you can say just like this and like this, only nucleus around, nothing. So that is known as chromophobe cell. And the chromophile cells are further divided into acidophil and also basophil. So acidophil is when they look with red or orange color, then they are known as acidophil. So there are some cells like red and orange, then it is acidophils, and some are blue color cells, then it is known as basophils when the granules stain basic dye with hematocylin. We use HND bases, right? So the basophil granules are also pars positive, and the acidophil cells are also called as alpha cells, and the basophil cells are also called as beta cells. And in electron microscopy of this acidophil and basophil, we mainly see the abundant dense coat vesicles in the cytoplasm. And next, both these acidophils and basophils are divided further into subtypes based on the size, shape of granules in them. And we correlate with the immunohistochemical methods like responsible for production of individual hormones to be recognized, and also functional types of the cells. So types of acidophils. Now let us see the types of acidophils. So mainly the acidophils are nothing but the somatotropes and the memotropes. So the acidophils and the somatotropes are nothing but forming the growth hormone, which is required for growth, and memotropes also lactotromes, which produce a prolactin hormone, it is important for milk production, and also are similar the growth and also female activity of mammary gland, and also during pregnancy and lactation. So this is all about that is milk producing and growth. So actually you can remember like acidophils are for growth, because growth hormone, and another growth is in female, that is for the milk production. So prolactin and somatotropes, so acidophilic. And next comes the basophilic cells. So the basophilic cells are having corticotropes. Corticotropes is the first one, and next the thyrotropes and the gonadotropes. Corticotropes acts upon our adrenal gland and it produces adrenal cortex, and it is pars positive and its cytoplasm is weakly basophilic, and it produces a corticotropic hormone. So this also produces a complex molecule known as proepiomelanocorticotropin, and it is broken down into ACTH and also other substances. So other corticotropin hormones that have been identified are also beta lipotropin, beta LPH, and alpha melanocyte, and also hormone alpha MSH and beta endorphin. So next, thyrotropes. As a name you can say thyroid gland. So it acts on thyroid gland producing thyrotropic hormone, also known as THS. And next, gonadotropes. Gonadotropes are also known as delta basophils. There are two types of hormones which mainly in male and female. So FSH, LH, right? FSH in female, it helps in follicle stimulation and also estrogen production in ovaries, and in male for the spermatogenesis. And in female we also have another thing, that is the maturation of corpus luteum and also secretion by the progesterone. It is called the luteinizing hormone. In the male, the luteinizing hormone also stimulates the production of androgens and also by the interstitial cell of testes, and it is also called as the interstitial cell stimulating hormone. So by this we are completed. And also according to investigation, two classes of gonadotropic hormones are produced by different cells, but other works also held that some cells can also produce both hormones. So now let us see the chromophobe cells. So we have completed chromophilic, that is acidophilic and basophilic cells. Now let us check out the chromophobe cells. So the chromophobe cells are mainly doesn't have any, not stained darkly, and there further details about so that is only about chromophobe cells, and they represent cells similar to various type of chromophils. They are also somewhat representing with the chromophils like memotropes, amotropes, thyrotropes, gonadotropes and corticotropes. Now let us check out some other details about pars anterior, that is nothing but our anterior pituitary. So in anterior pituitary, somatotropes constitute 50% and memotropes constitute 25%, corticotropes 15 to 20%, gonadotropes only 10%, and somatotropes are mainly located in the lateral part of anterior lobe. That is important. Laterally somatotropes are present, and thyrotropes are concentrated in anterior part, median part, and the corticotropes in the posterior part, median part, and gonadotropes and memotropes are mainly in the throughout the anterior lobe. So gonado and memo are throughout the anterior lobe. For example, this is what they are saying. For example, this anterior lobe, right? So gonadotropes and memotropes are like all over the anterior lobe. But as I said, laterally we have somatotropes and also thyrotropes are in the anterior and median part. So by this condition, and corticotropes are in the posterior part and also in the median part. So the size and dense cord vesicles are highly variable, and about 400 nanometers in somatotropes and 300 nanometers in memotropes and 250 to 700 nanometers in corticotropes, 150 to 400 nanometers in gonadotropes. This is about dense cord vesicles. Well, let's see the pars tuberculosis. What is it? It is consisting mainly of undifferentiated cells. So some are acidophilic and some are basophilic. And next, pars intermedia. If you remember pars tuberculosis, as I showed here. So this is a pars tuberculosis. Right now we have discussed about it. Now let us check out pars intermedia. So the pars intermedia is poorly developed in a human hypophysis. Not so important in human, but let us check what are there. It mainly conspicuous in feature, so it contains mainly colloid filled vesicles. So they actually look like this. These are known as colloid filled vesicles. They look like this, and these are remnants of actually red case pouch. And also the beta cells are other secretory cells like chromophobe cells are also present, and pars intermedia produce mainly the melanocyte stimulating hormone, that is nothing but producing the melanin pigment, and also increase pigmentation of skin, and also other produce ACTH, adrenocorticotropin hormones. Endorphins are also present in the cytoplasm of secretory cells. And the secretion of hormones from the adenohypophysis is mainly under control of hypothalamus, as we understood. So now let us check out neurohyperfaces, that is a posterior pituitary, also known as pars posterior. It is mainly having the numerous unmyelinated nerve fibers. This is important point, and which are the axons of the neurons mainly located in the hypothalamus, because the hypothalamus neurons are only controlling this pars posterior. So those neurons only come here. And most of these nerve fibers arise mainly in the supraoptic and paraventricular nuclei. So this, as I already discussed in physiology, neuroanatomy, I think everyone know. And situated between these axons, there are also supporting cells of special type known as pituocytes. So pituocytes are the supporting cells present in between them, and they have a long dendritic process which lay parallel to the nerve fibers. And the axons descending into the pars posterior from a hypothalamus ends in a terminal closely related to capillaries, because they need to send the hormones to the blood and to reach the target organ. And the pars posterior of hypophysis is also associated to release the blood into blood of two hormones, that is vasopressin and ADH. So actually remember, anterior pituitary almost produces all types of hormones, but the posterior pituitary only mainly produces two types of hormones, that is our ADH, antidiuretic, also known as vasopressin, and also the oxytocin. So only these two hormones are produced by the posterior pituitary. So posterior oxytocin, right, it is mainly for the contraction of smooth muscle of uterus and also of mammary gland. And ADH, it acts as a reabsorption of water by kidney tubules. So now these are synthesized in neurons located in supraoptic paraventricular of hypothalamus. So vasopressin is mainly present in the supraoptic nucleus, and oxytocin is mainly present in the paraventricular nucleus. So these are bound with a glycoprotein called neurophysin, and they pass down to the axons of neurons concerned through the infundibulum and pars posterior. Here they are released into capillaries of region and enter the general circulation, thereby reaching their end organs. So blood supply, how the blood is supplied supplied by superior and inferior branches from the internal carotid arteries, and also some branches from anterior and posterior cerebral arteries also supply, and the inferior hypophyseal arteries are distributed mainly to pars posterior, and the branches from the superior set of arteries are mainly supplying the median eminence and also the infundibulum. So end in the capillary plexus, and from there the portal vessels arise, and this portal vessels descend through the infundibular shock and end in a sinusoids of the pars anterior. If you remember, in pars anterior I showed the sinusoids, so there they end, and the sinusoids are drained by the veins and end in a neighboring venous sinus. Of course, every blood ends in drains in the vein. So we'll notice that it is unusual that two sets of capillaries are intervening between the arteries and veins. So one of this is the median eminence and also the upper part of the infundibulum. In this we can find this type of thing. And the second set of capillaries are represented by the sinusoids of the pars anterior. So this arrangement is referred to as hypothalamo-hypophyseal portal system. Hypothalamo-hypophyseal portal system. So the vessels which are mainly descending through the infundibular stalk are easily damaged in severe head injuries. So this leads to loss of function in the anterior lobe of Hypophysis cerebral. So this is a question like whenever there is a head injury, which lobe is mainly injured? That is the anterior lobe of pituitary gland is mainly injured. That is pars anterior is most commonly injured when there is an head injury. So next, control of a secretion of hormones of adenohypophysis. So how they are controlled? The secretion is mainly under the control of hypothalamus, as we know, and those especially in the median eminence. So as I said, median eminence is also a part of pars posterior, right? So you can see median eminence is also a part of pars posterior. So the median eminence plays an important role in the conduction of nerves which are passing through it. And next comes, and also in the infundibular nucleus. So the actions of these neurons end in relation to capillaries in the median eminence and in the upper part of the infundibulum. So as I said, all these hormones need to reach capillaries. Which point they actually end in the capillaries is mainly at the point of infundibulum and also at the point of median eminence. And the different neurons produce specific releasing factors for each hormone of adenophysis. So of hypothalamic and releasing factors we can study it in histology, and they release into capillaries, and the portal vessel arise from the capillaries and factors to the pars anterior of hypophysis. So they stimulate the release of appropriate hormones. Some factors inhibit the release of hormones and synthesis and discharge releasing factors by the neurons concerned in under nervous control. So whenever required they will release, and whenever there is a negative feedback or positive, according to this negative and positive feedback actually our endocrine system works. So whenever there is negative or positive feedback, according to that sometimes it may inhibit the hormones or sometimes it may release the hormones according to the body level of hormones. So they serve as an intermediate between the nerve impulses and the hormone secretion. So they are referred to as neuroendocrine transducers. So some cells called tantites present in the ependyma may also transport the releasing factors from neurons into the CSF or from CSF to the blood capillaries. So methods play an important role in the control of adenohypophysis. So recent studies also indicate that the circulation in relation to hypophysis may be more complex, like the entire neurohypophysis is from the median eminence of the pars posterior is permeated by continuous network of capillaries in which blood may flow in either direction only, and the capillaries provide a route through which hormones are released in the pars posterior can travel back to the hypothalamus and also into the CSF. Remember this point. And one more is that some veins are draining the pars posterior pass into adenohypophysis, and secretions by the adenohypophysis may thus be controlled not only from median eminence but also by the entire neurohypophysis, right? The next, the blood flow in the vein connecting the pars anterior and pars posterior may be reversible, producing a feedback from adenohypophysis to the neurohypophysis. So by this we complete our hypophysis cerebri or our pituitary. Next let us start with the pineal gland. Back to channel. Now let us check about our pineal gland, that is part of endocrine histology. So pineal gland, first its function is to mainly produce a melatonin, and it controls the circadian rhythms, that is our wake and sleep cycles. So that is its main function in our body. Now we need to know about its structure and histology. So it is a small pyriform organ which is mainly related to the posterior wall of the third ventricle of the brain, and it is also called as the Hypophysis Cerebri. And it is first known as vestibular organ, that is no functional important, but now it is also considered as endocrine gland of the great importance. That's why we are studying it. So the mass of cells amongst which there are blood capillaries. So you can say these are the mass of cells among which we have the blood capillaries. Why have blood capillaries? Because it need to produce the hormone into the blood capillaries. And also the nerve fibers are distinctive feature of pineal gland in such sections, and the presence of irregular mass made up of mainly calcium salts, mainly it contains calcium salts, and the masses constitute the corpora arnica. So corpora arnica is nothing but the masses of calcium salts forming an irregular mass. It's known as corpora arnica. You can see this is a corpora arnica, and these are the nuclei of Pinalocites and also the neuroglial cells. So these are neuroglial cells and nuclei of the Pinalocites you can find in the histological section. And next is that this corpora arnica is also known as brain side. This is a characteristic feature of a pineal gland in a histological section, and the organ is covered mainly by connective tissue that is representing the pia mater. So pineal gland is covered by an organ that is pia mater capsule, and from which the septa passes into the interior. So from that only the septa passes into the interior. So now we can see here show the features of Pinalocite. Now let us check out it. So organ is made up of main cells, that is a Pinalocite. So this is each Pinalocite, and each cell has a polyhedral body, right? You can see the polyhedral body and containing a spherical oval irregular nucleus. Yeah. And next, the cell body receives a long process with expanded terminal buds. So expanded terminal buds are present, so it receives long process. And next, that end in relation to the walls of a capillaries, because they need to produce a hormone, or in relation to the ependyma. Why with ependyma? Because it also releases into the CSF sometimes of the third ventricle. So the cell bodies of the Pinalocite contain both granular and also agranular endoplasmic reticulum, and a well-developed Golgi complex. So organelles are present, and also mitochondria, right? Many mitochondria, that is important. An organelle of unusual structure made up of groups of microfibrils and perforated laminae may be present. That is known as caniculate lamellar body. It is actually an unusual organ mainly, and having a perforated lamellae, many groups of microfibrils are present. So the process of Pinalocite mainly contain the numerous mitochondria. Apart from other organelles, the terminal buds, as I said, terminal buds, they also contain the vesicles, which are mainly having the monoamines and also polypeptide hormones. And the neurotransmitter gamma aminobutyric acid is also present. And the Pinalocites are separated from one another mainly by a neuroglial cells, and that resembles the astrocyte in structure. So remember, this is the neuroglial cells, so they are separated mainly by neuroglial cells, and it is in the shape of a astrocyte. And next we have the nerve fibers which are present mainly in the pineal are sympathetic nerve fibers, that is of mainly adrenergic, and they are unmyelinated and release the pineal secretions which appear to require the sympathetic stimulation. So sympathetic stimulation is required to produce the pineal secretions, and the Pinalocites produce mainly number of hormones that are chemically indolamides, polypeptides, and the hormones have important regulating influence, mainly chief inhibitory. They inhibit mainly other endocrine organs. So organs which include are adenohypophysis. So adeno anterior pituitary is inhibited, neurohyperfaces posterior pituitary, that is both pituitary gland is completely inhibited by our pineal gland, and also thyroid is under it, parathyroid, adrenal, cortex, medulla, gonads, and pancreatic islets. So whenever we have pituitary problem, so we may also have this problem because it may inhibit them. And the hormones of the pineal body reach the hypophysis both through the blood and also by CSF. So the pineal hormones also influence the adenohypophysis by inhibiting the release of many hormones. Adenohypophysis produce many hormones. So if the pineal is affected, of course adenohypophysis is affected, and many hormones reaching our body will be affected. So the best known hormone of pineal gland amino acid is melatonin. Melatonin also causes changes in the skin color in the amphibia. So the last concentration of melatonin are present in the pineal gland. So considerable amount of 5-hydroxytryptamine. Everyone know that is nothing but serotonin. So first the serotonin converts into the melatonin in a pineal gland, and which is a precursor of melatonin. So the presence of related enzymes is demonstrated. So synthesis of discharge of melatonin is mainly influenced by exposure of animal to light. So actually there is a pathway like, so whenever light falls on the retina, it enters light. So whenever light comes, it gives signals to the pineal gland, and less norepinephrine is produced, then less melatonin is produced. In darkness, the pineal gland is mostly active. So the pineal gland is most active in the darkness, that is more melatonin is produced. So the neurological pathway concerned involve hypothalamus sympathetic nerves, and because of the light mediated response, pineal gland may act as a kind of biological gland, biological clock, which may produce a circadian rhythms, that is variations following every 24 hours in various parameters. So it is suggested that suprachiasmatic nucleus of the hypothalamus play an important role in a cyclic activity of the pineal gland. So actually first light from the retina enter into the suprachiasmatic nucleus, and the suprachiasmatic nucleus actually initiates the paraventricular nucleus, and from the paraventricular nucleus it goes into superior cervical by our spinal cord lateral horn, and from there it gives message to the pineal gland where it converts the serotonin into melatonin. So this is a process by which our pineal gland produces melatonin and it affects us. So the cyclic activity, the nucleus uses fibers from the retina, right? It projects into tegmental reticular nuclei located in the brainstem, and next the reticulospinal fibers arising in this nuclei influence the sympathetic preganglionic neurons located in the first thoracic segment of the spinal cord. So these axons of neurons reach the superior cervical ganglion, as I said the process. So from there from where the nervus corani arises and supplies the pineal gland. This is important. Nervus corani arises and supplies the pineal gland. And it also been stated that the pineal gland degenerates with age. The corpora arnica was considered to be sign of degeneration of a pineal gland. But now recent studies are saying that the organ does not degenerate with age, and the corpora arnica is now regarded as byproduct of active secretory activity. That is, now the recent studies say completely opposite. So it has been postulated that polypeptide hormone first exists in the form of complexes with a carrier protein called neuroepiphysin. So when hormones are released from the complex, the carrier protein combines with the calcium and is deposited as the brain sand, because calcium deposits is nothing but our brain sand known as corpora arnica. So this is about the pineal gland, and next we will discuss about the thyroid gland. Now let us check out thyroid gland histology. So thyroid gland is mainly covered by a fibrous capsule around it, and it is having a septa which is mainly extending into the gland, by means of which it is divided into lobules. So lobules are in a thyroid gland, and microscopic diagram of each lobule is mainly having an aggregation of follicles. For example, you can take this as each lobule, and this is actually each lobule. In this each lobule you can say aggregation of follicles over here. So each follicle is mainly lined by the follicular cells. So you can see these are the follicular cells, and that rest on the basement membrane. So the basement membrane lying on the basement membrane, these are the follicular cells. And next, the follicle is having a cavity, homogeneous material colloid filled with colloid. So this is a homogeneous material filled with colloid, and it is pink in a hematocyanin and eosin stain, and the spaces which are present between the follicles are filled by stroma. So this is the stroma, and the stroma is containing the delicate connective tissue over it. And also there is also numerous capillaries. You can find some of the capillaries, and also lymphatics, and the capillaries lie in close contact with the walls of the follicles. Why? Because follicle having this colloid, and hormones are produced, and that hormones are very close to produce into the capillaries, because it is a endocrine gland. Into the blood, endocrine gland releases the hormones. So it is in close contact. And next, the thyroid gland contains C-shaped cells. So you can see these are the C-shaped cells. These are known as parafollicular cells. These are intervened here and there. They are scattered, means they are not having a perfect location, like they are always scattered, and between the follicular cells mainly. Yeah, see, this is one follicular cells. So between the follicular cells there are parafollicular cells. As the name you can say parafollicular, in between the follicular cells. And they also lie in the basement membrane. and they may also lie in the intervals between follicle. And connective tissue stroma surrounding the follicle contain a dense capillary plexus, lymphatic capillaries, and also the sympathetic nerves are present. And you can see these are different variations. This is moderately active, like here you can see the cells are like in the corner. This is highly active and this is inactive. If it is inactive, you can see there is more amount of collagen present in a follicle. And next let us see the follicular cells. So follicular cell vary in shape depending on the level of activity, as I said. So normally average at the level of activity means average level of activity, there are cuboidal cells. So moderate, these are cuboidal cells, when the cell is completely inactive, then they are flat and squamous. You can see flat and squamous when it is inactive, and when it is containing a evident colloid. And lastly, when the cells are highly active, they have a columnar cells and a colloid is scanty, means less colloid, but columnar cells are present all over the follicles, and the different follicles may show different levels of activity according to the different variations. So now, the follicular cells mainly secrete two hormones which influence the rate of metabolism. That is, iodine is essential for these hormones. So one hormone contains 3 atoms of iodine in each molecule. Then it is called as T3, T4. 3 means triiodo, means 3 iodines, and you add one more iodine, then it forms T4. T4 is also known as thyroxine, remember, and T3 is much more active than the T4. And the activity of the follicular cells is mainly by thyroid stimulating hormone, TSH, which is mainly produced from our Hypophase Cerebral or hypothalamus, and it indicates many activity increased by the sympathetic stimulation also. And next, let us see in the electron microscope, each follicular cells having a presence of apical microvilli, right? You can see here the microvilli, and there are also evident granular. You can see granular endoplasmic reticulum over here, and also it contains a prominent supranuclear Golgi complex, right? Golgi complex, granular, and also lysosomes, microvilli, and the secretory vacuoles. So all this is a structure of a electron microscopy of a follicular cell, and you can find out the many secretory vacuoles present in the apical part, right? So why these are present, what let us see. So the synthesis and release of thyroid hormone takes place in 2 phases. So the first phase is Thyroid Globulin. It is a glycoprotein which is synthesized mainly by a granular endoplasmic reticulum. So granular endoplasmic reticulum forms a Thyroid Globulin, and it is packed as usually by our Golgi complex. Golgi complex always helpful in packing and forming the secretory vacuoles, and the vacuoles travel into the luminal surface where they release the Thyroid Globulin, and then into the follicular cavity. So this Thyroid Globulin, this is a follicular cell, right? So the follicular cell we see in detail. So it releases into this colloid material. So what it releases, it is nothing but the vacuoles travel luminal surface and release a thyroglobulin. Thyroglobulin is produced by endoplasmic reticulum. So it is released into the cavity. And what happens? Mainly this process takes place by exocytosis. So here the thyroglobulin combines with the iodine to form a colloid. So the colloid is filled with the iodine. It is ionated thyroglobulin. So in the second phase, what happens? The particles of the colloid are taken back into the cell. So first this cell only releases the colloid, but again this cell is only taking the iodine back into the cell by endocytosis. So within the cell, the ionated thyroglobulin acted upon by enzymes like lysosomes. Here you can see lysosomes. So acted upon by lysosomes and releasing the hormone T3 and T4, which pass basally through the cell and release into the blood. You have seen that follicular cells lie very close to a capillary, so they release into the blood. So this is how T3 and T4 are released into blood. So the hormone is produced in the thyroid gland, mainly T4, and the output of T3 is less. But in liver, kidneys and other tissues, T4 is also converted to T3 by removal of one iodine, of course. So T3 and T4 are circulating in blood, are bound to the protein thyroxine binding globulin, TBG. So the bound form of hormone is not active, remember. So this is the process by which we can see here the iodine is absorbed from the blood and also glycoprotein synthesized in the follicular cell, right? Both together reach the form the monohydrotriazine and dihydrotriazine. Both together form in the colloid, they form iodinated thyroglobulin. Again this is taken back into the follicular cell by endocytosis and they form the T3 plus T4 released into the blood. So now let us see the parafollicular cells, also known as C cells. So these are clear cells and they are also known as light cells. They are polyhedral in shape. They are having the oval eccentric nuclei and they are typically lying between the follicular cells, as I said, on their basement membrane. And they may also be adjacent to the follicular cells. And remember, they do not reach lumen like them. These cells follicular cells, they're reaching the lumen, but parafollicular doesn't reach the lumen. And then some species, many follicular cells may lie in a connective tissue between the follicles and are arranged in groups. And in EM, we can see well-developed granular endoplasmic reticulum, Golgi complex, numerous mitochondria, and membrane-bound secretory granules all are present in parafollicular or C cells also. They secrete the thyrocalcitonin, which is completely opposite action of parathyroid hormone. So whenever calcium is increased in our body, these parafollicular cells are released and decrease the calcium level from the bone. So this is achieved by suppressing bone resorption by osteoclasts. So C cells share features of APUD system. We are going to see that further and are included in this system. So further for development and anomalies of thyroid. So this is we can see in the embryology. So this is the complete histology of thyroid gland. Next let us check out the parathyroid gland. Now we will learn about the parathyroid gland. So the parathyroid gland is mostly related with our thyroid gland. So hence the name parathyroid, means very closely related to our thyroid gland. It is having the superior and inferior lobes on either sides. Like related to parathyroid, there are totally 4 in number. That is, for example, thyroid glands have 2 lobes divided middle by isthmus. So this side we have upper one lobe and upper one parathyroid, lower one parathyroid. On the same time, on the left side also we have upper and lower. So total 4. Sometimes it may also reach up to 8 parathyroids. So it is also a connective tissue capsule made up of connective tissue capsule, and it is extended downwards to form a septa, and it enters into the gland substance, and the gland has a network of reticular fibers. Within the gland, we will find the many network of reticular fiber which support these cells, and also there are many fat cells known as adipocytes, we know. And next, it's also present in the stroma. These adipocytes are present in our stroma of a parathyroid. And the parenchyma of the gland is mainly made up of, it is arranged as cord-like structure. And also there are numerous sinusoids. You can see numerous white spaces, gaps over here. These are known as sinusoids, which lie in the close relation to the cells. And the cells of the parathyroid are mainly of 2 types. That is the chief cells, which are also known as principal cells, and the oxyphilic cells. So you can see there are most number of pink cells, chief cells, only nuclei is seen, like you can see almost this blue color totally filled is a chief cell, hence the name, because there are many in number. They are eosinophilic chief cells are principal cells, and the oxyphilic cells are eosinophilic. You can see here some pinkish cells or red or pinkish cells. These are known as eosinophilic cells or oxyphilic cells. These are less in number when compared to the chief cells, and chief cells are numerous than the oxy cells, and the latter are absent in the young. That is, the chief cells are absent in the young and appear a little before the puberty. And next, the light microscope of the chief cells are seen to be small rounds and with vesicular nuclei. And the cytoplasm, you can see it is clear, either mildly eosinophilic or it may be basophilic. That is about our oxyphilic cells. They may be sometimes mildly eosinophilic or mildly basophilic. Oxyphilic cells only appear only after the age of puberty, but not in the young. And next, the cell accumulates glycogen and lipids and also looks clear. That is about oxyphilic cell. And in contrast, oxyphilic cells are also much larger and contains the granules that stain strongly with acid. Sometimes they are also granules. So the nuclei are smaller and stain more interestingly than those of chief cells. And there are 3 types of chief cells, mainly light, dark and also clear. They have been stained. So cells intermediate between the chief cells are also been present. And the oxyphilic cells are also being described. So now let us see the chief cells produce the parathyroid hormone, which is also known as parathormone. Hence the name chief cells. Chief cells, you can remember, they are chief because they are many in number and they are also producing a hormone, so they are chief cells. And this hormone tend to increase the level of serum calcium. So whenever the body's calcium is decreased, they increase the calcium. That is increasing bone resorption through stimulation of the osteoclast calcium, and also increase the calcium resorption from the renal tubules, and also they inhibit the phosphate resorption, and also enhance the calcium absorption from the gut. So in the electron microscope, we can see the active chief cells, which are mainly having abundant granular, because as they are active, they are having the abundant granular endoplasmic reticulum and also well developed Golgi complex. So as Golgi complex is present, there are small vacuoles are formed, and especially in the parts of the cytoplasm near the edges and blood sinusoids we see them. And the features become much less prominent in an inactive cell. So in active cell only these features are more, and both active and inactive cell contain glycogen, amount of which is greater in inactive cells, because glycogen it is stored in inactive cells. And the normal parathyroid the number of inactive cells is greater than the active cells. In the electron microscope is seen that the granules of oxyphilic cell are really mitochondria, large numbers of which are present in the cytoplasm. And the true secretory granules are not present, and the function of oxyphilic cells is still unknown. So only chief cells we know, it produces a parathyroid. So this is about our parathyroid gland. Now let us see about suprarenal glands. Now let us continue with the suprarenal gland. It is nothing but the adrenal gland, as the name suprarenal, upon our kidneys. So these are actually, as we have two kidneys, even these adrenal glands are also two in number, that is right and left suprarenal glands. They lie in our abdomen and they are corresponding to the poles, superior poles of a kidney. So yeah, these are also known as adrenal glands, and they are covered by a connective tissue capsule, and that connective tissue capsule only divides into septa, as we discussed till now, into the gland substance. That is, septa extend to the gland substance, and two functional distinct parts are the superficial part called as cortex, as every organ has a cortex, outer cortex, and an inner medulla. So the volume of cortex is actually 10 times more than a medulla. So let us understand first about cortex. Next go to medulla. Actually most of the cells are present in the cortex which are producing different kinds of organs. So let us see in details. So what are the layers of cortex? The cells are arranged in a form of cords. So you can see in the cortex like this. Here all cells are arranged in a form of cords, and there are sinusoids, like somewhat spaces intervening between these cords. And the basis of arrangement of cortex is divided into mainly three layers. So what are those three layers? So you can see here the one is a capsule, and the second is a zona glomerulosa. This is a zona glomerulosa layer, and the third one you can see here, this is a zona fasciculata. This is the largest layer, and the fourth one is a zona reticularis. You can see like reticularis network like structure, this fourth layer, and the fifth one is a medulla, and the sixth one is a sympathetic neurons over here. So this is medulla. So first let us understand about the cortex. So in the cortex only we are having the different layers. So as I said, the capsule is made up of connective tissue capsule, made of connective tissue. So second layer is nothing but outermost layer of our cortex is the zona glomerulosa, and it is it's like inverted U-shaped cells. You can see, right? This is inverted U-shaped cells are seen here, and they are forming echinus-like groups and constitute the outer 1/5 of the cortex, right? This is the outer 1/5 of the cortex. So next, second layer is the zona fasciculated. It is the biggest layer. You can see it is covering almost this much space. Here the cells are arranged like straight columns, right? Like straight, yeah. And two cell thick, yeah, it is thicker. And the sinusoids intervene between the columns. Yeah, some white spaces you can see, those are the sinusoids. And the layer forms the middle 3/5 of the cortex. So this is middle 3/5, that is 1/5 only. So the second layer is a zona fasciculata. Next let us go deeper, that is the innermost layer of the cortex is nothing but occupying the 1/5, that is zona reticularis. So this is the zona reticularis. It is because it is made up of cords that branch and anastomose with each other and also form a reticulum. So as anastomosis they are forming reticulum. This is reticularis. So in light microscopic structure, if we see cortex, mainly the cells of the zona glomerulosa are seen to be small, polyhedral. So yeah, in this picture we can see. So the cells are small, right? And they are polyhedral, and also columnar cells are present sometimes with the basophilic cytoplasm and deeply staining nuclei. Yeah, you can see the nuclei are very embossing out here, right? In the first layer, that is zona glomerulosa. Now let us see about zona fasciculata cells. So these are large cells, polyhedral, and with the basophilic cytoplasm and having the vesicular nuclei. This is important. And next, these are like important points to remember. And next, the cells of the zona fasciculata are very rich in lipids, so which can be demonstrated by suitable stains. But because of the tissue processing, what happens is they are emptied and they appear like a vacuolated appearance. And they are also sometimes containing small amounts of vitamin C in the fasciculata. This is important. And next, let us see the cells of reticularis. These are also similar to the fasciculata, but the lipid content is less over this reticularis, and the cytoplasm is eosinophilic. Yeah, somewhat red you can see because of eosinophilic, and the cells often contain a brown pigment. So here the cells are containing the brown pigment, and here the cells are also containing vitamin C some amount. Here the cells are U-shaped, you can inverted U-shaped. So this is about cortex we completed, and let us see the electromicroscopic structure of the cells of the cortex. So you can see this is the electromicroscopic structure of a cell, each one cell of a cortex.
In all layers of the cortex, that is characterized by the presence of abundant agranular smooth endoplasmic reticulum. So you can see abundant of smooth endoplasmic reticulum, that is especially agranular. You can see no granules over it, just smooth endoplasmic reticulum. And also we have a Golgi complex, right? And Golgi, remember that Golgi complex is only best developed in the Zona Fasciculata. So only in this layer we are having a rich amount of zona. In fasciculata only we are having rich amount of Golgi complex. And next, the mitochondria are elongated in the glomerulosa. The mitochondria are elongated in the glomerulosa and spherical in the fasciculata. Remember this thing. So mitochondria is elongated in glomerulosa and spherical in fasciculata, and fasciculata, one more point, is Golgi complex is abundant. And next, unusual with the tubular cisternae instead of usual plates in reticularis. So in reticularis, our mitochondria is usual plates actually should be, cisternae should be tubular usual plates, but it's unusual with tubular cisternae instead of usual plates in the reticularis. So that is important. So the mitochondria is different in different layer, that is important. In the first layer the mitochondria is elongated. In the second layer the mitochondria is spherical, right? So first elongated, next spherical, next tubular cisternae. Actually in mitochondria we have normally usual plates are present, but here it is containing tubular cisternae and reticularis. So every layer is having different type of mitochondria. So this we can find in the electron microscopic study. So what are the hormones produced by a suprarenal cortex of our adrenal gland? So mainly the zona glomerulosa forms a mineralocorticoid hormone, that is nothing but aldosterone and a deoxycorticosterone. So what these hormones are doing? So the glomerulosa hormones are influencing mainly electrolyte and water balance, and also secretion of aldosterone is influenced by renin system, which is formed by juxtaglomerular cells of the kidney. So we know this, and the secretion of hormones is mainly by glomerulosa, appears to be largely independent of hypophase cerebral. So actually it produces on its own, it doesn't require any message from the hypophysis. And next, the cells of fasciculata are producing the cortisone, that is a glucocorticoids, and also cortisol dietary. What it does is widespread, including on carbohydrate metabolism, protein metabolism, and also decrease the antibody response to anti-inflammatory effect. And next, fascicularis layer, that is the biggest layer, it produces small amount of dehydroepiandrosterone, that is DHA, which is an androgen. And next, reticularis. Reticularis produce glucocorticoids, sex hormones, estrogens and androgens. So the suprarenal gland is essential for life, because it is forming different types of hormones. So whenever it is removed or it is destructed, then it leads to death of the organism, unless the hormones are produced or supplied artificially, right? Because these are very important hormones. So increase in the secretion of corticosteroids causes dramatic reduction in the number of lymphocytes. So suprarenal cortex is very important. Now let us see into deeper structure, that is the medulla. Functionally and embryologically, the medulla is distinct from the cortex, right? Because the gland is fixed in whenever it is for example, if the gland is fixed in a solution, like in a salt of chromium, example in potassium chromate, dichromate, if we put this medulla, then the cells of medulla are showing some yellow granules. So what are those granules called? That are known as chromaffin reaction. That is, the cells give positive reaction, and these are called chromaffin cells. So the cells of the suprarenal cortex are not having this reaction and these granules, right? So the chromaffin reaction and chromaffin cells are only belonging to the medulla. It is a special character. And the medulla is made up of group of columns of cells, and the group of columns are separated by a by wide sinusoid. And the cells are columnar, polyhedral and also basophilic. And functionally the cells of suprarenal medulla consider to be modified post-ganglionic sympathetic neurons. Like typical post-ganglionic sympathetic neurons secrete the noradrenaline, norepinephrine and adrenaline epinephrine in the blood. So the secretion takes place mainly when adrenaline, all this in fear, stress, whenever we are in anxiety, etc. So this leads to stimulation of sympathetic nervous system. Sympathetic means it increases the heart rate and also blood pressure is increased. So an electron microscopic study, if you see the adrenal medulla, so this is a EM of an adrenal medulla. We see abundant granular endoplasmic reticulum, right? Abundant granular endoplasmic. So if you remember, in cortex we are having smooth endoplasmic reticulum, but here we are having granular endoplasmic reticulum. And also there is a prominent Golgi apparatus which is present, and also the cells are mainly containing the membrane bound secretory vesicles, right? And also the some cells of these vesicles are small, electron dense, while others are large and not dense. So former are containing non-adrenaline and later are containing the adrenaline. So the suprarenal medulla is now included under APUD cell system in contrast to suprarenal cortex. The medulla is not essential for rest as its functions can be performed by other chromaffin cells too. As you know, cortex only having many hormones. So medulla only chromaffin cells, chromaffin reaction, and difference is here granular endoplasmic. It is means not only regulating blood pressure, this heart rate, that to increasing the sympathetic activity. It is not useful in any other hormones, so it is not so useful. So whenever it is removed, there is no problem because the same function can also be performed by the chromaffin tissues. So now let us see some other organs are also having endocrine functions like the paraganglia. What is it? It is an aggregation of cells which are mainly similar to the adrenal medulla and they are found collectively referred to as paraganglionic and present in relation to autonomic ganglia. So the name paraganglia, and they give the positive chromaffin reaction and they receive the paraganglionic sympathetic innervation and also have the secretory granules containing mainly catecholamines in their cytoplasm. So the cells of adrenal medulla paraganglia are believed to develop from the cells of neural crest cells and they are richly that is having vascularized and also they act as endocrine glands in alternative sites for production of mainly catecholamines in the fetus and also in the early postnatal life. And the adrenal medulla is yet fully differentiated because of their histochemical and ultrastructure features. Cells of paraganglia are also included under APUD system. So what is this APUD system? We are going to see it in further. So most of the paraganglia retrogress with age but some persist into adult life. So cells similar to those of paraganglia are also present with some sympathetic ganglia and they are called as SIF or small intensely fluorescent cells and they are believed to act as a interneurons. So now para-aortic bodies and carotid bodies are also sometimes included in the paraganglia. Now let us see what are these. Para-aortic bodies are two elongated bodies which are present mainly each side of the aorta near the origin of inferior mesenteric artery. You know inferior mesenteric artery comes from the aorta. So at that site mainly this para-aortic body is present and they are like two masses may be united to each other or may be separate and they are connected. If they are united, they are connected by a band passing across the aorta. And these bodies have structure mainly similar to that of a adrenal medulla and the cells secrete noradrenaline. You know, yes, medulla secrete noradrenaline, so these also secrete and the aortic bodies also retrograde with age. Next, carotid bodies. You know, this is a small oval structures present on one side of the neck at the bifurcation of common carotid artery. We know common carotid artery is divided into internal carotid artery, external carotid artery. So at that side there is a carotid body and near the carotid sinus. Actually, carotid sinus is present towards division of internal carotid artery, right? So between them, actually the carotid body is present. So the main function of this carotid is to act as a chemoreceptor, that is to monitor the oxygen and carbon dioxide level in the blood. So reflexively control the rate and depth of respiration centers and also through respiratory centers which are located in our brain cell. So in addition to this, the carotid bodies are also believed to have an endocrine function. So what is that? They contain a network of capillaries in intervals which there are several types of cells and the carotid bodies have a rich innervation also. So the most conspicuous cells of the carotid body are known as Gloma cells. So what are those? Here in this diagram you can see these are the Gloma cells. These are the most conspicuous cells of type 1 cells and they are large cells. They have several similarities of neurons like they have dendritic process, the cytoplasm contain membrane bound granules with neuropeptides and also has prominent and the carotid body has the most peptide present in it, that is encephalin and also present include dopamine, serotonin, catecholamines, VIP and also substance P are present. So the cells are in synaptic contact with the efferent nerve terminals of the glossopharyngeal nerve and the chemoreceptor impulses pass through these fibers to the brain. And some Gloma cells also show the synaptic connection with the endings of the preganglionic sympathetic fibers and also with other Gloma cells. So the organization of endoplasmic reticulum in them shows the similarities of mainly nissl substance. So they are surrounded by sheet cells and resemble neuroglial elements and also similarities to neurons. So the possibility that the cells also release a dopamine substance. So they are sometimes described as neuroendocrine cells and also having a nervous connections. And the exact significance of glomas is still unknown and understood at present. So they could be sensory receptors to oxygen, carbon dioxide, tension, and also dopamine released by them influence the sensitivity of chemoreceptor nerve endings, and also they may serve as an interneurons. So at part of Gloma cell, other cells are also present like carotid bodies. So what are these? These are having sheet cells or type 2 that surround the glomus, and few sympathetic, parasympathetic, postganglionic neurons, and endothelial cells of blood vessels, muscle cells in the walls of arterioles, some connective tissue cells, carotid bodies richly innervated like afferent nerve terminals from glossopharyngeal nerve, form synapse with Gloma cells, preganglionic sympathetic parasympathetic fibers end on corresponding ganglionic cells, some preganglionic sympathetic fibers end by synapsing with Gloma cells, postganglionic fibers arising from sympathetic and parasympathetic nerve cells with carotid body innervate muscles in the walls of arterioles. And the precise mechanism by which the carotid bodies respond to change in oxygen carbon dioxide tension is not understood. So it is not certain to which cells or nerve terminals are responsible for the function. So this is just like oral reading because this is some extra points which is not of that significance. So last is the diffuse neuroendocrine or APUD cell system. So what is this? This is also apart from a discrete endocrine organs, considered to be a group of endocrine cells scattered in a various part of body, so present all over the body, and they share a common characteristics with our with each other and also with the cells of some discrete endocrine organs. So the cells take up a precursor substance from the circulation, they process them by decarboxylation to form the amines or peptides, and therefore included in the system known as APUD, that is amine precursor uptake and decarboxylation cell system. So these peptides or amines serve as hormones, and many of them act as neurotransmitters, and APUD is also called a diffuse neuroendocrine system, and the cells of this system are spherical oval membrane bound granules with dense core, and there is an electrolucent halo around the dense core, and these cells are of give positive chromaffin test and also known as chromaffin system, however appear to closely related to the cells and that are not chromaffin, and the tendency to consider all the cells under the common category of diffuse neuroendocrine cells. So this system is regarded as representing it is a link between the autonomic nervous system and also classically recognized endocrine on the other hand, and because of this having both features. And the effects of amines or peptides produced by the cells of the system are like local, like neurotransmitters, and sometimes widespread like those of hormones. So list of cells included under APUD as well as the secretions is large. So discrete and are now included with the heading. So let us see other. There are various cells of adenohypophysis, neurons in the hypothalamus, synthesize hormones of neurohyperfaces like oxytocin, vasopressin, and also synthesizing releasing factors controlling the secretion of hormones by adenohypophysis, and chief cells or parathyroid producing parathormone, and C cells, parafollicular cells producing calcitonin, cells of adrenal medulla like secretin, adrenal and noradrenaline include SIF cells of sympathetic, and also cells of gastroenteropancreatic endocrine system include cells of islets, insulin, glucagon, amines, endocrine cells scattered in the epithelium of stomach, intestine, and also 5-hydroxytryptamine, that is serotonin, glucagon, dopamine, somatostatin, produce p-mortlin, gastrin, cholecystine, secretin, vasoactin, intestinal polypeptide, VIP, and some other peptides. So Gloma cells of carotid bodies produce dopamine, noradrenaline, melanocytes of skin producing promelain, and some cells in the pineal gland, placenta, modified myocytes of heart called the myoendocrine cells, and renin producing cells of kidneys. So this is just like orally reading because not so important. So by this we complete our endocrine system. We understood histology of endocrine system.